All right, welcome everyone. Thanks for being here today. I'm sure we'll have a few more people joining us in the next minute or two, but we'll go ahead and get started. My name is Renee Bell. I'm GFI's Senior Science Content Specialist, and I'm filling in for Elliot today, who is on a well-deserved vacation. I'm also joined today by Amanda and Isabella, two of our scientists from GFI Brazil, who will be providing technical support during the Q&A portions of our call today. So thanks to them for joining us. For anyone who is joining this call for the first time, we hold these industry-wide calls every month for any company that wants to participate in the cultivated meat value chain. The purpose of these calls is to introduce and welcome new solution providers to the industry with the end goal of establishing new collaborations that will accelerate R&D and commercialization. If your group is interested in presenting on a future call, or if you're working with a company that may benefit from being on these calls, please feel free to put them in touch with either me or Elliot directly. I do have a few housekeeping items to share with you today, and I'll put the links in the chat afterwards, since I'm no good at multitasking. You may have heard the Good Food Conference is taking place in September, from September 18th through 20th in San Francisco, and the program is fully available online. It looks fantastic. We're really excited about it. And if you can join us, you'll want to register by August 31st to avoid the next rate increase. You can join us in person or virtually, and I hope to see you there. Also, a reminder that applications for GFI's 2023 Research Grants Program are due September 21st. This is for projects that are up to 2 years in length and $250,000 in budget. And yes, we do fund proposals from companies, and we love to see companies involved in collaborations. So please see our RFP for full details. I'll be dropping that link in the chat. And finally, the UN FAO is collaborating with the China National Center for Food Safety Risk Assessment to hold a stakeholder roundtable meeting on cell-based food production and precision fermentation on the 6th of November, 2023. A call for presenters is now open, so please review the detailed selection criteria before you apply. And the deadline to submit that application is a week from today, August 31st, which is hard to believe we're approaching the end of August. So that's it for today's announcements. Today, I'm delighted to welcome Romy Dalton from Cocoon Bioscience and Eduardo Agustin from Sticta Biologicals. Both will be talking about their technology platforms for producing recombinant proteins and other ingredients for the cultivated meat industry. So during the presentations, please feel free to drop questions in the chat, and we will get to them at the end of each presentation. Romy, I'll pass the floor over to you.
Thank you. Good morning, everybody. And first of all, thank you to the Good Food Institute for inviting us this morning here to this webinar and present you our company and our thrilling technology. So what I would like to talk about in the next 20 minutes is regarding this question here. So how come, if we have known for a long time now that growth factors are the key bottleneck for the cultivated meat industry to be able to scale, why it hasn't been solved yet? Well, we think the key is in the cost of production of these growth factors using the conventional technologies available today. So we at Cocoon, believe that it is possible to produce these growth factors at a lower cost with a still sustainable platform. So our mission is to catalyze industries that are bottlenecked due to this high cost of production of some of the starting materials. So let me introduce you Cocoon, first of all, our story and the milestones we have achieved and are about to achieve. So the idea of using living insects as natural bioreactors to produce recombinant protein was generated back in 2005 by a veterinarian who funded a company called Algenix. So in the following decade, this company was launched. The proof of concept of the technology was successful, and a pilot scale automatization was even developed. After that, from 2015 to 2022, the technology was consolidated by a full patent protection of the technology, and also an EMA regulatory approval of the first animal health vaccine produced in this technology. By the way, just to let you know that I joined Algenex as a COO in 2015, and therefore I witnessed the consolidation of this technology. So it was in that very moment when Cocoon was created in order to explore this thrilling technology in other applications. So Algenex still uses it for animal health, and Cocoon was created to exploit other applications. So since it was created in late 2022 and until 2024, what we aim to do, well, what we have achieved already is a 15 million euro round. We have set up an R&D laboratory, and we are currently in this second half of 2023 setting up and constructing a pilot and an industrial facility in order to reach the market leadership in 2025 in basically two target markets. So both have in common that they are bottlenecked by, as I was saying, the high cost of producing some of the starting materials. So one is the healthcare enzymes, which are enzymes involved in the modification of nucleic acid that then our clients use in gene therapy or mRNA vaccines, for example. And the other one, which you would be more interested in, is the cultivated meat industry. And as I was saying, the growth factors that bottleneck the scaling of this industry. So let me first explain you how this technology works. So as any other recombinant protein technology, we get some starting materials and introduce them in an upstream process where protein gets produced. And then we extract and purify these proteins through a downstream process, and then just a fill and finish standard process. So in our case, the starting materials are two. Our technology is based on the baculovirus technology, which has been used by industry since the 90s, where you use a virus that only infects insect cells and that you can genetically manipulate to harbor the gene that will express the protein of interest. And then the other starting material you need is a host cell. So we also use this recombinant baculovirus that the industry uses, but instead of using bioreactors to cultivate the insect cells, we substitute this by the cocoon stage of the natural host insect of this baculovirus. So we at Cocoon are self-providers of both of these starting materials. So in the case of the insects, what we do is we maintain a population of this insect, which is called Zygoplusiani. So starting with some moths, which is the adult stage, they lay eggs. From these eggs, a larvae are hatched, and these convert into chrysalises, from which an adult emerges again and starts the cycle all over again. So the production of these eggs is so efficient that you, with a small size population, you get enough eggs to feed this process and maintain your population, and also to feed the pupae production process, which is basically the same, but you stop the cycle in the pupae stage, which is our starting material. So we go to the upstream by basically automatically inoculating the recombinant baculovirus into the pupae or chrysalises. We incubate from 3 to 6 days, only controlling temperature and humidity. And in this period of time, the protein gets produced. So you can store that biomass frozen until you're ready to go to your downstream process. So what you do is a conventional downstream process for purifying this protein, this produced recombinant protein. Conventional downstream just after a first step where you convert this solid biomass into a liquid extract by crushing the pupae. Then you go to conventional, totally conventional technologies. In all our downstream processes, we include a 0.2 microfiltration step to make sure the product is sterile, because as you can imagine, using living beings, you cannot have a sterile process from the beginning. And then you just go to a normal conventional fill and finish, depending on what you decide, the format in which you decide the product. So all the process is animal-free quality, and you can run the process under GMP conditions if needed, as it will at one point happen with the healthcare enzymes in our case. So let me talk you about the advantages of this platform. Well, the most important, as I was saying at the beginning, is that it makes the process cost efficient. So we are able to produce a recombinant protein at a much lower cost. And this is made basically by the conversion of the other three advantages, which are simplicity, linear scalability, and speed of development. So let me walk you through each of them. So simplicity, well, if you compare to bioreactors, and you think that your bioreactor is a natural bioreactor, a cocoon, which is already doing what it normally does in nature, which is expressing every single gene in the virus that gets to infect it, you realize how simple it is. The only things you need to control, as I was saying, is temperature and humidity to make sure that the environment of these pupae, while they are producing the protein, are at a constant temperature and humidity. You don't need to control any other factors such as pH or gases, etc., which makes things much simpler, as you can imagine. This reduces as well the risk of cross-contamination or contamination in general, because the production is being produced is happening in this naturally encapsulated environment, which is the cocoon. And finally, it's a very modular platform, which gives a lot of flexibility as well to it. Second is linear scalability. Well, it's linked to simplicity as well, but it's also linked to short development times, because you don't need to validate when you want to change your scale. All you need to do is just get the eggs that your insect population are producing and decide with only 2 weeks in advance how many pupae you would like to inject in that particular week. So depending on the size of pupa biomass you need to inoculate, you just use more or less quantity of eggs. And you get either a low or a high scale production, making this very flexible, simple, and also shortening the development times. So typically we run a proof of concept for a new protein in around 3 months. We then go to develop the industrial scale. Well, we develop the downstream for that particular product, and we scale it to industrial scale. And in 4 to 6 months, and with 3 more months, we can get the product in the market. So basically, in roughly 1 year, you can get your product in the market from scratch. So all these advantages make, as I was saying, this process very cost efficient. So when you think about the small space you need to run the platform, the reduced amount of equipment and more simple equipment than when you use a bioreactor, you realize there's a much reduced capex. Also, it's highly automatized and therefore makes the OPEX lower as well. And finally, it's very low cost maintenance. It's very simple and low cost to maintain. So this is to show the portfolio of growth factors we are generating. Well, we have the typically used in the cultivated meat industry, such as FGF2, PTGF, EGF, and we are currently developing others that are more complex to develop, such as TGF-beta1. And we produce these in any species that the client might need, bovine, human, porcine, whatever. As long as we have the sequence, we can potentially express any growth factors the industry might need. We obviously have tested these growth factors. We need them to be cheap, but also active. So here's an example where a leading life science company has tested one of our growth factors. In this case, it's a lyophilized bovine FGF stored at 4 degrees. So they performed a percentage of proliferation of cells at different quantities, using different quantities of this growth factor in comparison with a control. And you can realize they work very similarly. This is a second example where we used the same growth factors, but we stored it at room temperature for a week before doing the test. So we make sure the stability is okay for shipment to the US, for example, from Spain. In the same way, the proliferation of the cells grows with a quantity of growth factor in the same way that in growth factor that's already in the market. This was performed by a university in South Spain. Regarding regulatory and quality, we have designed the process so it can meet both the FDA and the EU guidelines, and both markets, food market and healthcare market, because they have different requisites. So we have built a process that's flexible and can meet all of them. As I was saying at the beginning, we can produce GMP grade products if needed. So which is our infrastructure today? Well, we have an R&D lab in Madrid, Spain, where we develop new products constantly and scale them up to 10 liters. We are currently in this Q3 setting up a pilot lab in North Spain, where we will scale up to industrial scale these downstream processes and validate them to get them ready to be transferred to the industrial facility, where we will produce at industrial scale. And this we plan to get it ready for early 2024. This plan will be a very flexible and modular production. As I'm going to show you now the characteristics. Well, it's a 4,000 square meter building that uses a bit more than a half for production. The starting capacity will be 5 kilos of recombinant protein, but because of the modularity of the platform, we can easily, in the same facility, ramp up to 20 kilos by adding downstream lines. So if you think about the small amount of space you need for such a big quantity of protein, you realize why I'm saying all the time that the capex is really reduced with this platform. The building is located in a biotech cluster up North Madrid, where existing customers from Cocoon already exist, so which is very convenient. And the upstream scale give us margin to even increase the capacity much more in the same building. So we are really making a very flexible industrial facility that can gradually increase the capacity to adapt to the increasing capacity or needs of the cultivated meat industry. So in conclusion, what we offer to the world is a novel expression system with insects as bioreactors that has been validated by our customers and that can offer a much reduced price of the growth factors for the alternative meat industry. And we are getting ready for the increasing needs that this industry will have in a short period of time. So thank you very much for listening. If you would like to know a bit more about us or the technology, I leave you here a QR to go directly to our LinkedIn and also our webpage. And then if you have any questions, I will be happy to answer those.
Thanks, Romy. That was a great presentation. We have a couple questions in the chat already, and I encourage others to either come off mute and ask their questions or add them to the chat. Eduardo, I will turn it over to you now.
Okay, thank you. Okay, so good morning, everybody, and good afternoon for some of you that should be in other continent. Sticta, I'm the CEO and co-founder of Sticta Biologicals, and which is committed to provide key ingredients for the cultivated meat industry. So we are a precision fermentation company situated in Santiago, Chile. And our mission is to accelerate the widespread adoption of cell-based meat, focusing particularly in creating a great taste and price parity compared to conventional meat. Here we are with Andres, my co-founder. This is me, Eduardo, and Andres. We are visiting our partner in Israel, Supermeat, with which company we have developed several products, and they have validated most of our products. And here we are tasting their cultivated chicken meat, which is a hybrid, either a roll here or a chicken hamburger. And our mission is to accelerate this and allow everybody in the world to taste excellent cell-based meat. To do that, our products are wrapped in a trademark name, which is a Smart Serum. And the Smart Serum is a combination of several components which could be delivered either individually or all together, and it's composed of key protein and growth factors, as well as nutrients. And our main advantages compared to our competitors is that we have reached a low cost. We have validated with Supermeat high performance in at least avian stem cells, and also great taste of resulting cells. So in order to do this, we have set up an experienced and complementary team that has already a history of success stories. So my name is Eduardo Agosin. I'm the CEO and co-founder. I have more than 30 years of experience in precision fermentation and a substantial track record of scientific publications in the field. And I'm joined by Andres Aristia, who is a business entrepreneur who has also a strong experience in international business in the agro-industrial sector. and also two minor partners and young bioengineers, Vicente and Sebastian, the CSO and CTO, respectively, with a strong experience in synthetic biology and bioprocessing and modeling. We recently complete our seed round and were able to raise almost $2 million that will allow us to consolidate our platform. And we are joined by Proveg International as well as family office Amarena. So how we do that? How we produce Smart Serum and the different components of Smart Serum? So we have a non-conventional food-grade yeast cell factory. It's not Pichia, by the way, that we have domesticated for biomanufacturing proteins and growth factors. So we program this non-conventional yeast cell factory. We optimize its production through transformation of different proteins, and we scale it very easily in bioreactors to produce the compounds of interest. Let me show you a little bit or describe a little bit in more detail the process. So what we have is a yeast cell that we have engineered to produce and secrete, which is one of the interests of this yeast cell factory, is that secretes an important, significant amount of proteins. And so we have engineered the yeast in order to produce insulin, transferrin, and also a few growth factors. The distinctive capacities of these factories that it's composed by a groundbreaking, fast-growing food-grade microbial cell factory. It secretes, as I said, the recombinant proteins to the medium, which allows to a much easier downstream processing, which is minimal, by the way, and so we get a food-grade protein at the end, and we have developed a chemically defined culture medium that we have shown is quite easy to scale, at least at till 50-liter bioreactors. So let me show you some of the proteins here. Sorry to show these gels, but just to prove that we really are producing these proteins and growth factors, here we are producing transferrin. Each of the columns here are different clones or different colonies of the non-conventional yeast cells. And here are the parental that do not produce, for example, transferrin. Same case here, different colonies producing insulin or leaf or oncostatin M. And also we can produce at least 2 or 3 simultaneously of these proteins. Here we have produced insulin here and transferrin, as you can see. This is the control. So let me, we have validated the proteins with super meat, and I think this is one of our distinctive advantages, is that we co-develop tailor-made solutions with our clients. In this case, super meat asked us to produce a chicken leaf, and we produce it in 3 months and send them from Chile to Israel, which was something, by the way, so they could test the pluripotency of the product we have produced in our platform. It's a food-grade sea leaf, and so we compare it here with a commercial purified leaf, human leaf, by the way, and we can see here with different stains or markers, nuclear stainings and markers that pluripotency in avian stem cells is preserved, and also there is no toxicity of the ultrafiltrated leaf, as you can see here that the number of cell density over time is very similar when you use a basal medium with the purified leaf, or the basal medium with our ultrafiltrated leaf. Another interesting, I think, advantage or capacity we have in our labs is the proprietary computational design algorithms that we are using to develop novel and particularly stable variants of key proteins and growth factors. I show you here some of these in silico developments that we are testing now for the bioassay. And particularly, we are developing single-chain insulin, which is particularly thermostable and also stable, more stable so with a higher stability in the culture medium. This is the model of our single-chain insulin compared to the commercial single-chain insulin available in the market. And these numbers here indicate that they are much more stable using this different mechanistic and artificial intelligence algorithms. Same thing for FGF2. We also check in our platform, in our bioinformatic platform, that there is optimal interactions of the different variants between the receptor and the proteins here. So here in yellow, you can see the model of the receptor. This is the active zone that is interacting with our model 38, in this case, the single chain insulin, and same thing for the FGF2. This is the protein FGF2. This is the active zone of the whole receptor for FGF2. So I think we have here a tool which is quite distinctive from the other companies working in the field. So we also produce a yeast lysate simultaneously or after we recover the proteins from the supernatant. We collect the yeast cell biomass and through a very simple process, we autolyze it, and we're able to recover amino acids in a soluble form, amino acid derived from proteins in the cell, as well as some vitamins and other interesting cofactors. And we separate them from the cell debris through a simple centrifugation. And we have tested this yeast lysate first in the proportion of amino acids and compare it to two of the best commercial yeast extracts in the market, according to Supermeat, that they have selected as the best extract for growing their avian stem cells, liver and oli. And you can see that we have similar quantity of amino acids. Around 40% of the whole yeast extract is formed by amino acids. And here this is the relative amount of the 20 amino acids. And there are some small difference, but all the amino acids are there and comparable to liver and oli. But furthermore, Supermeat has shown that our lysate has no toxicity and a higher performance than other yeast extracts in the market. Here, they have done in duplicate in 2-liter flasks the different passages using our lysate, and you can see here that after each passage, the cells recover very quickly and apparently there is no toxicity according to them. They have last for more than 20 passages, by the way. But also compared to the competitor, the cells grow 25% faster, as you can see here, after day 8 than other yeast extracts. Now, why Sticta is also your best option? Because we have low prices today, and we are producing either recombinant proteins, bovine insulin and bovine transferrin at prices really competitive, as well as some growth factors. And we can develop any other protein or growth factors with very performant titers or productivities that and very fast, maybe less than 3 months, that we can provide to our clients according to their necessities. And also we provide the yeast lysate at a price comparable to the prices in the market, around $50 per kilogram. Our products, our proteins are food-grade proteins, and we can offer more than 80% purity of our products. So to do all this, we could not do it alone. And here is the space of Sticta. We have partnered with Proveg, and a company working more in the cell bio, with an expertise in cell bio, Merkin, in Santiago. And we have several collaborators like Extracellular for bioassays, Protera for a French company from artificial intelligence, protein design, Bioconsulting, a very interesting company for the technico-economic analysis. Melissa, we are working very hard on identifying bottlenecks in the metabolism of our cell factory using proteomics. And also Lawyer Buffet for IP strategy, Atova for the registration of our first insulins at the FDA, and GPC Bio and others as CMOs. And our clients today and collaborators that we are working with is Sifi Foods, Upside, and also, of course, our first company and strong collaborator, Supermeat. So just to finish, this is our team in the fermentation lab. We have a platform of 4 1-liter bioreactors where we optimize the process at the fermentation lab level, and also we could scale here to 50 liters. So as I told you, we are committed to accelerate the widespread adoption of cell-based meat. Thank you very much. If you want to know more, please contact us. I give you here my email.
Wonderful. Thanks, Eduardo. It's great to learn more about your work. Well, I do thank everyone for joining today. And if you have any follow-up questions, I'm sure Romy and Eduardo would be more than happy to field those via email. And I do hope to see many of you at the Good Food Conference next month. And if I don't see you there, I'll see you at next month's seminar, which will be held on September 26th. So have a great rest of your day. And thanks again for being here. And thanks again to our presenters.
Thank you. Thank you, Renee, for the opportunity.
You're very welcome.
Thanks a lot, everybody.
Bye-bye.
Bye.